So my name is Jarrod Medeiros. I'm the product manager for E-WorkBook here at IDBS. Been working with Roland in particular and ChemAxon for quite some time. I realize that based on some of these presentations, I am a biologist in a room full of chemists. So please take it easy on me. I'm going to give a little bit about how we're working with ChemAxon on the biology side of the house. So with E-WorkBook, historically, people know E-WorkBook as an ELN, and it started out really as an ELN software, but it's really grown with IDBS to become a platform. Started out as an on-premise, desktop-based application platform, and now we've built that up into a cloud-based, web-based application platform as well. And historically, we've always been known as sort of the biology-based ELN, and we've, in the past couple of years, been working with ChemAxon to build in a number of chemistry tools to really step up our game in the chemistry space. And I'm not going to talk about those today. I have colleagues, and I'm sure you can see webinars and things. We've been promoting those for quite some time. But I want to talk a little bit about how we're leveraging some of the tools from ChemAxon in the biology space to really advance that in our web-based platform. Now, with E-WorkBook, we have a few different modules that I'm going to talk about today. In addition to the ELN, which I think everybody's kind of familiar with what an ELN is and what that does, we have our advanced, which is sort of our advanced spreadsheet technology. Historically, this was sort of known as BioBook. This is where we do a lot more LIMS-like functionality with structured data management and things like that. And we have our inventory module, which is where we do more of the inventory tracking for equipment, samples, materials, and those types of things. Now, all three of those modules are actually impacted by the biology functionality, and we're leveraging the ChemAxon technology to be part of that. Now, historically, with E-WorkBook, we've done a lot within the BioProcess space, and this is kind of standard biomolecule production. So working in the cell line development space to develop the cell lines that are going to be used as the expression system, and cell culture and purification, and all of the surrounding activities that go along with that. These are areas that we've been focused on for years and years now, and we've done a lot of work in this area. But one thing has always been lacking within our platform, and that's been proper biologics registration. And this is where we're working with ChemAxon to work with the new web-based technology and really provide something unique and different from a biologics registration perspective. Now, thinking about the biologics landscape here, we have a few areas, starting with nucleotides, moving through to the expression systems, which are typically cell-based when you're talking about standard biologics, sometimes more on the synthetic side, as well as the final product, typically, unless you're working with siRNAs and these types of things, but the peptides. Peptides could be, I'm sort of lumping those together, but they may be antibody-drug conjugates or any types of products of the biological sequence. Now, with this process, or with each stage here, there are a lot of things that go into this process as well. The materials that are used at the various steps, as well as the samples that are generated at each of these. And this is where we're tying E-WorkBook together with the ChemAxon technology to really provide a tool to cover that full ecosystem. And as you're working through that process, throughout the drug development life cycle, there are a lot of different process steps that have their own nuances. And this is where I really want to focus our effort with E-WorkBook is, aside from just the molecules themselves characterizing, storing, and being able to search on these, it's about how those fit into the overall process. Because especially with biomolecules, the process can actually have a major impact on what you actually produce. So with the BioProcess, for example, post-translational modifications, things that can happen to those molecules during the expression, all of these things are heavily impacted by what materials have gone in, what the process conditions are, these types of things. And at any one of these stages, there may be various levels of characterization that's going on. So you may not actually understand any of that until sort of later in the process. So what we want to do is be able to bring those tools, bring that kind of functionality to the end users and integrate it directly into the process as they're working on it. Now, when I talk about BioReg, I want to touch on something here, and I think actually the tissue presentation kind of already started this. But really, when we're talking about BioReg, there's sort of two aspects to it, right? There's the physical entities, and these are the samples, these are a vial of primer, the things that we can hold in our hand that we actually store. This is what we're actually working on at the lab bench at our daily aspect. And then there's the virtual entities, and that is the sequence of that item, and these are inherently related, right? So that the physical entities actually have some relationship to the virtual entities, but those each have their own relationships within. So if I bring in a cell line from an external vendor, and then I use that cell line to express a number of different supernatants, and then I take those supernatants and put them through some different processes, there's a whole relational aspect along the lines of those entities as they've gone through that at the physical level that may not be actually tied to the virtual level. By the same token, at the virtual level, if I create a plasmid that has a certain insert for DNA, that encodes for a specific protein. So there are sort of relationships along both of those that we're looking to track. Now, how we're leveraging the ChemAxon technology with IDBS is that we're using our inventory system as managing the physical aspects, and we're leveraging the ChemAxon biomolecule toolkit to do the virtual aspects. And we're using the toolkit and the BioEddy components to really serve as front-end and functional components of this functionality. Now, in an example scenario here, let's say we bring in a cell line from a vendor that has some properties to it. It's been transfected. It has something that we want to express. We then plate that out and clone it and generate a number of different clones to do that expression. And we may then sort of pare that down and scale that to however many clones we think are our best clones. Maybe do some additional processing to those. And then finally, we have our final clone that we express for our drug substance at the very end there. Now, in this scenario, there are a few areas where we might actually do a biologics registration in the virtual sense, and that is at the beginning stage and at the end stage. But all of these stages in between actually require that sort of physical genealogy, that sort of physical interrelationship to be able to really understand what's actually going on in the production of this, what's happened to those cell lines, what's happened to those as they've gone through this. So this is where at IDBS we're trying to sort of tie those two together and bring those to the end user so that they can have all of that data in one system. Now, with the workbook, historically we've done sort of the standard biologics in e-workbook, and here's an example of, say, a plasmid. But what we've also seen is that these sort of standard tools, there's a bit of a gray area with what's biologics and what are closer to small molecules from a characterization perspective. And so leveraging the Helm technology, as we're bringing our biofunctionality to the web, we're looking at using sort of the standard biology types that we've used in the past, augmenting that with the BioEddy functionality to be able to do some more of these other types of molecules and have all of those within one system. And then at the end, have that built right into the user's notebook. So this is an example of what we're working on right now with e-workbook, and this is the first phase of our integration with the ChemAxon tools here. This is bringing in the BioEddy functionality directly into e-workbook, leveraging the monomer sets, everything that Roland's talked about before and shown that, doing that right within your notebook. So the end users have this in their notebook, and they can actually if I go back to this one, you can actually see here on the right-hand side we have the registration panel, so they can edit, they can manipulate, they can add in their biomolecules directly into a workbook and register them from there and have those as part of their write-up directly in the ELN. So this is covering the sort of ELN side of it, that initial write-up side of it. But then what we're looking at doing is bringing that together with the inventory module. And this is what we're doing later in the year. So here's an example in our inventory module of a set of host cells that we might have in the workbook inventory. And then I can bring that directly into my notebook as well, showing this either directly in the notebook experiment itself, or if we're doing more structured data in our advanced technology. So this is using our advanced spreadsheet capabilities to actually do a transfection experiment and make those relationships to say, well, this plasmid and this cell line were transfected to produce this expression system, and then I'm going to use these downstream in further experiments. So tying all of those together as the end user is working them into their process. And that's really the key that we're focusing on, is not just making it so that end users have another system that they have to work on. I have to go into the biologics registration system and register this, but really integrating that into the end user process so that they're making these connections, they're entering this data, and they're tying these things together as part of their daily workflow. So it's all sort of built together, and this is, in my opinion, how we get the end users to really use these systems best, right? Because any of these systems, without proper data in them, if they're not sort of properly used, or if users are sort of half-heartedly using them and only entering once in a while, that really impacts the quality of the system. So from my perspective, making this as usable as possible, putting it in the end user's hands, and building it right into their process is kind of where we're going with the ChemAxon technology. And this is all being built using the BioEddy functionality and the biomolecule toolkit, the Helm standard as a standard backend from a biologics perspective as well. And what that allows us to do is cover chemistry, biology, and everything in between, all within one system. So leveraging the ChemAxon tools from a chemistry perspective, using our standard biology tools, and bridging that with the BioEddy and the biomolecule toolkit, sort of providing one platform that can cover all of those for the end user's needs.
What sort of release date? When are you going to be delivering it?
So the phase 1 release date is mid-year. So the functionality in the e-workbook as a notebook with BioEddy built into it and the integration to the ChemAxon registration system, that's coming mid-year. By the end of the year, we're looking at having the inventory system integrated as well.